Kids are supposed to be running around outside now. Right? Happy that it's raining. If you're from Portland or Memphis, you don't get quite so excited about rain. So we're having a few technical snafus. I transferred my talk to PDF, but I wanted to actually do half of my talk live using a web service called Gene Network. So I'll probably switch at about 12, 15 minutes. I would like to switch back and forth to this web service. The presentation, I'm giving it for my first time as a PDF. I think it'll work. I don't have too many fancy transitions. It's a little bit distorted, but the idea is we'll be introducing you to strains that you've already heard from Hanno and others, the canonical mouse, C57 Black 6, and the oldest inbred strain of mouse, DBA2J, were used to make a family of derivatives. Those derivatives are referred to the BXD panel, and I'll show you what you can do with about 40 years worth of data from the BXD family of strains. And we'll talk about this in the context of replicability, reproducibility, and refinement and improvement of data. So I need to explain what a recombinant inbred strain is to you. I went to the Jackson Laboratory in 1994 to get to learn about mouse genetics. I was a neuroscientist before that point, and Ben Taylor, one of the inventors of the recombinant inbred strains, gave a presentation, and after his presentation I had the foggiest idea what a recombinant inbred strain was. It took me about another 10 years to really understand them. Basically, I think the easy way to think about it is as a cloned family, where every member of the family comes in a pair, a male-female pair. So for Rob Williams, there's a Roberta Williams, and we can continue to propagate infinite. And we have, so here you can see some of the very nuclear families, BXD 49, 43, and we have about 150 of these percolating in the lab. About 100 of them are ready right now to rock and roll from the Jackson Laboratory. You can see one of these is named 73B. Turns out there's a 73, a 73A, and a 73B. They're all more related than they should be because of screw-ups during the breeding. Results in some interesting population substructure. But fortunately now we have statistical methods that make it pretty easy to clean up some of the breeding errors. What are they good for? They're great for generating phenomes, and I'll explain what I mean by a phenome to you in a second. There aren't very many phenomes out there in the big bad world. If you ask, how much data do we actually have for any one human, or how much data do we have for any one mouse, it's still pretty pathetic. Black 6 would probably be the reigning champ of all organisms on the face of the earth, except for maybe certain lines of E. coli and yeast. Certainly for mammals, C57 Black 6 would be it in terms of the amount of data we have. But we don't have even data across a population. And what we've tried to do is to achieve relatively even phenotype data across the BXDs. And we've done that as a community over a 40-year period. John Crabb got this started in behavioral genetics in the early 80s or late 70s, actually, when they were just coming online. So you can generate phenomes. Obviously, you can generate the genotypes for these animals. That's not a problem anymore. You can study complex causal interactions. Just forget about the genetics. You can say what correlates with what. You can replicate genotypes. So you can do GXE, GXL studies to your heart's content. And you can do it with a stable experimental paradigm of population, again, for generations. The really cool thing about this is I can take John Crabb's 1981, 82, 83 data, and I can reanalyze it, and it gets better because all of the context for his data is now much richer. And I'll show you how that works. The mapping is also much better. When this work was started, it was a major motion picture to get genotypes. We now take that for granted. So it's like a good wine in that way. It keeps getting, data gets better with age, and I don't think there are very few other cases where that's true. Usually data degrades with age. So what is the architecture of a recombinant inbred set? Fouad will tell you a little bit about another recombinant inbred set called the collaborative cross. The DBA, this particular cross between Black 6 and D2 was started by Ben Taylor in the early 70s. Black 6 and D2 don't really need any introduction. Hanno did that beautifully. They differ on many, many phenotypes that you heard about. Some phenotypes they don't differ by in terms of the phenotype, but the underlying architecture of the genetics is still highly different. So you can expose that. And you expose that by a meiotic recombination process. You take mom and dad, you cross them, you make the isogenic F1s. You then have the miracle of meiosis, which randomizes chunks of chromosomes, shown here in green and red. This is what, when you took genetics in high school or college, the F2 is what people talked about, the F2 and the backcross, that's what Mendel used. We're randomizing the chromosomes. So this goes back to Fisher's agricultural experiments in terms of extracting causality from that randomization as part of that extraction of causality. The problem with the F2s is you can't accumulate a phenome for an F2 or the diversity outcross. It's very tricky because you can only kill the animal once. We need to be able to kill animals multiple times or study them to be kinder and gentler. If you then take an F2, you put, again, a male and female in a cage together, and you inbreed them for 20 generations, you end up with a recombinant, recombined mother and father, hence recombinant inbred strain. It doesn't have anything to do with recombinant DNA in the sense of 1980s, 1990s. It means we've recombined chromosomes. So here's our randomization process. We have 150 of these. We know exactly what their genotypes are. We know exactly what's segregating in those genotypes. Can we predict their phenomes? Can we go from genotype to phenotype, from genome to phenome? That's the challenge to us. That's essentially the challenge of personalized medicine. If we can't do this for a bunch of damn mice, where we can study 100 of each, where we can do GXL, GXE, what hope is there actually to do personalized medicine for us? I'd say pretty piss poor. So until we can do this for mouse population or rat population, we should be very careful of our claims to be doing this for humans. So that's the architecture. Fouad will tell you a little bit about a much more complex situation. What's actually going on in this cross? So you understand the red and the green, but what does that actually represent in terms of sequence differences? It represents more sequence diversity than in this room. And I'm saying that because I don't see anybody from Nigeria here in the room. Probably about less than 5 million common sequence variants segregating this room. These animals, these two, just one family is segregating for 5 million common sequence variants. They're common sequence variants by default because we're dealing with the family. They're not really common, probably in mousedom. But from the point of view of genetics and statistical power, they're common. Roughly minor allele frequency of 0.5, which is optimal for doing a t-test or a NOVA. We now know exactly what's going on in this cross because both parents have been deeply sequenced. We've got about 120x for the DBA2J, and of course, Black 6 is a reference strain that we compare everything to. So we can take those 5 million sequence variants and we can categorize them. We can say we have so many missense mutations, so many nonsense, so many splice sites, so many frame shifts, so many copy variants. This is all specified. And the very cool thing about this is that recombinant inbred strains were used for classic forward genetics, going from behavioral variant down to gene. But now you can just as easily flip it. You can say, hey, I've got a variant in catechol-methyltransferase, or DRD2. What do I see up there in mousedom? So you can do reverse genetics, I would argue, more efficiently and at a better scale than you can with knockouts. And you can do it in a more realistic environment because you're looking at a genetically diverse family. So at this point, it really is the phenome stupid. A few years ago, I thought it was extremely presumptuous to call this the post-genomic era. And it really is kind of presumptuous to talk about it as the post-genomic era. It's really the phenomic era now, I think. That's what we see in studies of human populations. We need to get these large electronic healthcare records. So, thank you. Terrific. So, we've been building this phenome since 1973. In behavioral genetics, it's really been built since the Oregon group got started using them in about '81. And this is what Gene Network looks like, and I'll be switching over to Gene Network, time permitting. But this is a website. You just go to it right now and see if you can crash it. It's one of the oldest websites in existence. It's been up and running in one form or another since 1994. is something called the Portable Dictionary of the Mouse Genome initially. You select a species. It doesn't just have mouse. It's got human, rat, Arabidopsis, et cetera, potatoes, whatever. We select phenotypes, and then we can put in a search term. For the BXDs, we've got an enormous amount of data. Phenotype data at the very top. Genotypes, so that's classical stuff that you would be interested in if you're interested in replicability. Genotypes, we take that as a given now almost. And then these are all molecular assay data sets. So for example, here it says liver metabolome, liver mRNA, liver proteome. We have some metagenomic data. So you can get that whole, that stack of omics data that people yak about all the time. So if we put in the word memory in that search screen, we get about 100 hits for memory. It's not really 100 independent studies. It probably boils down to maybe 20 independent studies that did something memory-like. This is some recent work by Andy Holmes and colleagues using the touch screen assays in the BXDs to study learning and memory. Just gives you a flavor. I think the take-home message here is have a look at how these phenotypes are described. This is not how the data came to us. This was a major motion picture to get this annotation this way. So you may not be able to read that, but it says central nervous system behavior, learning and memory, colon. So that's sort of the controlled vocabulary. Discrimination test, correct responses using a touchscreen assay in a 3-month-old male. Supplementary trait data not in paper. So we've spent a lot of effort. I have to give credit to Alyssa for getting this started. She either took data out of the literature by using calipers and rulers and a bunch of undergrads, or in some lucky cases, like John Crabb sent us a big Excel spreadsheet with about 200 or 300 traits in it. We dumped it in a MySQL database. We did a lot of cleaning up, annotation. You can get pretty much the raw data. It's not the raw data. It's not the individual data. It's the strain means typically, because that's all you can get out of the literature for the most part. But this gives you an example of what the phenotypes will look like if you go into Gene Network. You can obviously download everything. You can add them to a shopping cart and do multivariate statistics, et cetera. So what does a phenome look like? These are classic phenotypes here, and that's 10 to the 3. So this is 3,000, 4,000. These are the strains of mice. And you can see that the phenome is inevitably uneven. Some individuals, you get dense data because they're easy to acquire, they're easy to ship. Everybody loves them. Others have poor breeding performance, or they died at some point, so you have sparse data for those. This is protein data and metabolite data. This is 10 to the 4. This is brand new quantitative proteomic and metabolomic data from Rudy Ebersol and Zamboni at the ETH. You can see we have very sparse data here on the original BXD sets from Ben Taylor. These are all the new ones that we've generated. We're biased in favor of our new ones because they're more highly recombinant, and they have pretty good classic phenotypes. So that's why those were used. But obviously, it's important not to end up with a too patchy distribution of phenome, or else you get in trouble when you're doing the correlations. This is mRNA. That says 10 to the 6, so that's a 3 million right there. These are mRNA assays, and that's a gross underestimate of what we actually have in there. So we have 240 expression data sets, mainly mRNA. Alyssa did the very first one on whole brain. The latest ones we put in there are from Ebersol of the proteomic data sets and metabolomic data sets. And in some cases, we have time series data, et cetera. Rutherford and stamp collecting. Yes, this is stamp collecting, but it's computable stamp collecting. And it's kick-ass. You can do things with this data set and play with it in a remarkably large number of ways. So that's the phenome. Just a quick plug. There's now a reverse method of mapping. Everybody knows about genome-wide association studies, I assume. You can now do a phenome-wide association study. You can say, I'm interested in catechol-O-methyltransferase. I've got the B allele and the D allele in half of my population. Now let me scan across the entire phenome and see what comes up with a high p-value, or I should say a low p-value. And here's the minus log of the q value for scanning the phenome. And the phenome is broken into phenosomes, if you will. behavior, CNS pharmacology, morphology, fat mass, blood chemistry, immune system, musculoskeletal. They get smaller just because of the way the community generates the phenome. It's very uneven. And so in our classification, of course, is also, there's nothing magic about how we've divided this. This is simply a heuristic way to handle the data. These are some traits that track to a particular sequence variant that we think is quite interesting. So more is different. I think Karl Marx said it first in the sense of income. There's a beautiful paper by Phil Anderson, goes back to a 1967 paper about emergent behavior that I recommend having a look at. This paper in 1967 ends, surely there are more levels of organization between human ethology and DNA than there are between DNA and quantum electrodynamics, and each of these levels can require a whole new conceptual structure. Beautiful statement. It's really true. I've been studying RNA now, mRNA, for 10 years. I still don't really understand. I don't think anybody understands the relationship between RNA and protein. There's a lot going on there. So each one of these levels is complex, and we have a long way to go. And getting coherent data is critical. All the data we've been talking about here is a little bit over there and a little bit over there. It's horribly confounded. You really can't process it statistically in a coherent way. With a reference population, you can. You can't do this in F2s. You can't do it in humans, really. So this is a figure that Alyssa prepared. Just shows what I mean by the exponential or multiplicative usefulness of every vector of data that you generate with the BXDs. So Hanno, if you can do it, in your group can do it with B and D, I will give you at cost 192 BXDs, and you can do whatever you want now across 24 genotypes, and it'll be awesome. Because every vector of data you generate, you can multiply it basically by 10 numbers of correlations. So obviously, if you've got 6 traits, you've got 15 correlations. And in our case, we have 3 million molecular traits, and we have 4.5 tera correlations, tera-Rs. So it gets terrific. Fisher, because he was a smoker, said correlation is not causation, although we know now he's wrong, and at least in the case of smoking, the cancer, that is pretty causal. We can turn a lot of these correlations into causality by using classic gene mapping. Gene mapping is a search for causality, and I'll show you how that works. I think I'll skip this in the interest of speed, but this just shows, well, I'll skip it, because I said I'd skip it. But cool stuff. So here's our stack of levels. Instead of just doing 2 strains, let's do 20, 30, 40, 50, as many as we can manage. And in our case, we've gotten up to a high of about 80 strains for some traits. So I want to switch over to the show and tell now. We were challenged by Philip to not just advertise. All of us have been advertising. I'm now not going to advertise. I'm going to show you. I'm going to show you the data if I can actually. I hate using Windows, but I'll try. If there's anything you can tell the dean about this room, make it work so that it works with the Macintosh. OK, so let's see. I want to switch over to just a browser. Oh, I might need my glasses too. How am I doing for time now? I'm OK. OK, so gene network, not gene network. .org. How many? 15, perfect, OK. That's where I wanted to be. So this is what gene network looks like. It defaults typically to a mouse. I hope you can see this, but it says species, group, type, data set, and then some search terms. And you can, I'm just going to leave it here, set for phenotypes. And in this case, I'm going to show you first demonstration. We're just going to have a look at brain weight. This is something that Doug mentioned. It was in the '99 paper. the famous or infamous 99 paper. So let's look at a trait that's in this paper by Hager that I just flashed through. I could search for behavior, and I'd get about 1,000 hits, in large part due to Alyssa's input, just a lot, but also it's just biased. There's just a lot of behavioral research in here. So Hager, let me make this well, I won't make it a little bigger. I'll just pray that you can read some of this. But with that particular search, for Hager, we have 13 records that are associated with that particular publication. And I'm going to sort them and take this one on the top, which is trait 12661. It reads central nervous system morphology, brain weight, repeat measurement prior to dissection, statistically adjusted for variation, sex, age, body weight, and for dissection error. Okay, let's actually look at the trait. So if I click on the blue link, I'm going to look at the trait. It tells me again what I have. It has a link to PubMed. I can put it in my shopping cart and play with other parameters if I wanted to. I have basic statistics. I can say, how many do I have? I have an N of 88, mean, standard deviation, et cetera. What does it actually look like? Whoops, I have some outliers. What am I going to do with it? Right? So this is a stuff that typically your novice, non-statistically inclined biologist doesn't know what to do. They're not even going to look for outliers in the first place. And they won't know what to do with the outliers other than throw them away. So here are the data with the error bars. Here it is, the same data now sorted from low to high. So mouse brain weight varies in the BXD family from about 380 milligrams up to about 500 milligrams. Nice data, has high heritability. If you computed the heritability, just the strain main effect. You can see that it's probably highly heritable just from the error terms there. The nice thing about the BXDs is I can almost convert any trait to have a heritability that's very high if I have enough budget, because I just resample. I'm interested in the strain mean. I'm interested in the difference between the strain means. Give me 100 strains and I can convert the heritability high enough to do whatever I need to do to get genetic correlations or genetic causes. So here's what the data look like. Univariate. Well, I'm not interested in univariate. I want to see, can I repeat? Oh, and here's another thing we do. You'll notice that some of these have been flagged with yellow. You have to prevent children from chopping off their fingers. And the yellow says, hey, be careful, there's an outlier here. Maybe you want to do something about it. So there I've sorted it from high to low. And now the data are not static. I can go in here and I can say, I'm going to Winsorize this data, and I'm going to call that 489. And I'm going to call that 489. And then to Winsorize this officially, I should do it in the symmetrical way at the bottom, but I really don't care because it's not a symmetrical problem. So I'm taking care of my outliers without throwing them away. I know that they're high. They're just too high. And now I can say, does it correlate with other measurements? And that's bivariate question. And I go to calculate correlations. You can see it defaulted to Spearman rank. Didn't really need to. But so I'm going to, now that I've handled the outliers, I'm going to go to Pearson product moment correlation. And I'm going to say, give me the top 500. But I could ask it to give me the top 20,000. There aren't 20,000, but I could ask it to do that. So let's do that. And let's see how the correlation, this is basically we're replicating the figure that John showed you in his talk, that figure, the correlation. So here it is. It's all calculated in real time. We don't want to pre-calculate anything because you see I tweaked the values. And now I can just do a search for crab. I think it's down here about 20. Yeah, here it is, 24. So here's the correlation with brain weight after a period of more than 2 decades. So Crabb 1992 versus Williams et al. Roughly 2002. That's a correlation of 0.85 with a p-value, nominal p-value of roughly 10 to the minus 7. I only did one test, so I don't really have to get too excited about correcting for it, but you can see we would. Okay, so that I would consider we've replicated that pretty well. We might be interested in why DBA2J is so much lower in John's data than it is in our data. What happened? Was there a spontaneous mutation? That's a pretty big difference. Everything else looks pretty stable. We could look for differences in the means rather than just the slope here. And this is pretty close. Well, I'm not sure what the slope is there, but all of this data is available. For you guys who are statistically savvy, go at it. You know, it's 40 years of data. It's all yours. You know, we're hoping that somebody will use it, and very few statisticians have used it. Let's see, I want to close this window. Whoops, I did not want to do that. Shit. Okay. All right, let's start again here in Hebrew. No, I got it. Don't worry, I got it. I'm okay. I'm just fussy. All right, here. Okay. That brain weight was Hager. The other one I wanted to show you is how you fish causality out of this. Okay, so the other thing we want to know is what's controlling that difference in brain weight. And we can go well beyond correlation, and that's the mapping functions. There are a couple mapping functions in here. There are actually a bunch more that we could put in, but I'm just going to use a very simple, essentially biserial correlation analysis developed by Haley and Knott. It has the advantage of being computationally very, very fast. So what I'm going to do now is when I push this button, I'm going to do a whole genome scan 2,000 times. And you'll see if all goes well and the computers at Memphis are working properly, we'll get our answer in about 10 seconds. If I wanted to, I could add a bootstrap test to estimate the precision in a sloppy way of the confidence interval. But I'm computing an interval map. So basically, I'm just asking, what's the correlation between the genotype and the phenotype? That's all mapping is. It's pathetically simple. The only issue is multiple testing, because you're doing many tests across the genome. You don't know exactly how many tests you're doing because of the complex genetic architecture. You can see that took almost no time. When we started this, Ken Manley was responsible and built the algorithms for doing this mapping. This would have been a 30-minute plus procedure. And that's why I don't care how complex a calculation is. I figure it's my job to the user to do it in under 10 seconds. And if I have to throw a supercomputer at it, that's my problem, not the user's problem. It doesn't matter how complex the problem is, in my opinion. We should be leaning into this. So chromosome 19, there's a QTL, quantitative trait locus. For those of you who don't know this stuff, on the x-axis are the chromosomes. On the y-axis is a chi-square statistic called the likelihood ratio statistic. Divide by 4, and you have a minus log p of the value. So this QTL has a minus log p of 5. It's pretty good. I call that its cause. We know it's causal kind of from the central dogma. DNA causes variance in DNA, causes variance in phenotypes. I'm pretty damn sure about it. Now it's just a matter of arguing which gene in the locus is actually responsible for the causality. The causality is given, at least when you have a QTL that's this strong, unless you did a screwed up in a royal way, it's fine. It's remapping it. It's stateless. It's dumb that way, but it's so damn fast that it doesn't matter. This is chromosome 19 showing you all of the genes on chromosome 19, showing you all the SNPs segregating the cross, megabases, showing you where our peak is. It's got this brutal architecture compared to those of you who've looked at nice, soft LOD score maps. It's brutal because we know exactly what's going on, down to like 100 kb or better in some cases. So I can now fish through here and say, what gene could this be? It might take me 10 years to figure out exactly what gene it is, but this is getting easier and easier. Okay, let's see, do I want to close that? No, I don't want to. I don't want to close you again. Close current tab only. Okay, let me go back to. I think you've got a feel for it. Again, there are about 4,000 classic phenotypes in there to play with, and millions of molecular traits and 5 million variants. Yeah. Do
you have an API?
We do. Yeah. So in Philip's thing, so here's the scriptable interface down here, data sharing, blah, 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 annotations. So if you just poke around here on the website. You can upload your own data. This is our version 1 of Gene Network. This started in 2000. The code for here, this is written in Python. The code started about 2000. It's a terrible, terrible mess. So talk about being embarrassed about code and not wanting to share it. It isn't a GitHub, but you have to be have very strong digestion to handle this code. We've refactored the whole thing using good modern architecture circa 2008, 2009. That's Gene Network 2. I'd be happy to show it to you. Let me just, so Gene Network 2 is, so here's the GitHub for Gene Network 1. Here's the GitHub for Gene Network 2. There's a cloud service. If you wanted to wrap it in Docker, you could do it. If you want to have a copy here in Tau, be delighted to give you a whole copy. And you can play with it and screw with the code and fix it. Here's what the Gene Network 2 version looks like. It's just a little, we're trying to make it look like NCBI so that when I die, somebody at MCBI will say, this is good enough, and it looks like NCBI will put it in. We'll see. So that's...
Rob, can I?
Yeah.
So I've never used the system, and
shame on you, Robert.
I know the principles only. I don't know the BXDs in terms of resolution. So do you think 100 strains?
Yeah, let me show you that. Actually, thank you. I'll give you your $1 afterwards for asking that question. But first, I have to get back to my talk. How do I get back to my talk?
I'll just tap.
Tab down here. That one right there. Okay. Here's my talk. Okay. So this resolution issue is a big bugaboo, and I think I left it in here. Okay. See? Next slide. Okay. So this is empirical resolution. This isn't bullshit resolution from simulations. What we did is we took 25,000 sissy QTLs. I'm not going to explain it to you, but you can ask you can ask the sissy QTL experts in the audience, and basically ask what's the offset between where I know the gene is, it's causal, and where the QTL peak is. And the worst possible QTLs you can map in the BXDs typically have a LOD score of 3, and those will typically have a precision of plus or minus 3 megabases. That's the worst you can do. That's with 67 strains. We now have twice as many. So worst scenario on a monogenic QTL, and that's also a kind of weird concept, but these are all monogenic QTLs. You can have a polygenic QTL, and it can get much uglier. But that's sort of the asymptotic resolution is probably plus or minus a megabase with the resources we have now. So it's good enough. So people that get real freaked out about precision, I think they're just barking up the wrong tree. I think it's a total red herring. We have enough priors now that we don't need, if you need more than a megabase precision, you're stupid, basically, is my opinion.
Because of the deep sequencing
What?
methods, you can cover larger distances with sequencing. So it's because of the molecular methods.
Yeah.
The length of DNA that you can take a look at is actually increased.
Right.
So you get less and less precision in linkage analysis.
Yeah. So I showed you this. I didn't show you this, but this is behavior. There are more than 1,000 traits in there. You can try it yourself. We can improve. I mentioned the data improve. I could take John's data from 82, 83, and I could remap it, and it could be kick-ass. I took some of Tamara Phillips's data that John did. Some awesome QTLs there now that weren't there when the study started. I pretty much introduced you. So let's look at I'm sorry Philip isn't here too. But how good is this in terms of what his sort of criteria were? How generalizable is it? First of all, it's a family instead of one damn strain. One strain does not generalize. So, bless you who are doing the informatics for the Black 6 UCOM, COMT, et cetera. That's good molecular biology. It has nothing to do with genetics, as far as I'm concerned. It won't generalize to DBA2J. So you have to incorporate. So in terms of things you cannot leave out, you cannot leave out genetic variation, or else you're just playing. You cannot leave out environmental variation because we're all subject to it. So those are things you must not omit. And unfortunately, you can't omit developmental noise. Maybe you don't want to. I don't know. Can you get the same animals' genotypes? Check. Yep, you can do that. They're all available from Jackson Laboratory. Can you get the code and play with it? Check. All open source, GPL version 3 of Faro license. Can I access the data and metadata? I showed you that. Check. Can I do the calculations myself? Check. You can download the data and you can do it in RQTL, whatever you want. Use a Bayesian framework for mapping. Can I correct for multiple tests? 50-50. So we've embedded it for the genome-wide scan. We have not embedded it for the phenome-wide scan. So we know how to do it, but we haven't implemented it yet as something that everybody can use.